Does anybody have anything to say before we start? All right. So we can start now, Shamash. So I run the kind of chemistry and life sciences rooms in Science Discourse, and every week that I present, just take a topic and give the basics of that. Today, we're going to talk about amino acids, peptides, and proteins. They're all related to each other, as we will see. So on slide 2, that's a cartoon of a cell where we have the cell membrane on the outside and the nuclear envelope or nuclear membrane inside the cell. And everything else that you see there, for the most part, except for the mRNA and the nuclear DNA that you see, everything else is a protein. So the receptors that are on the cell surface, they are proteins. Many of the signal molecules that hit the proteins are polypeptides. And signal transduction pathways inside the cell are all mediated by proteins in the sense that proteins do a reaction on other proteins to change their chemistry, and that way they send the signal down the chain. So we have a ton of proteins in our bodies, and how much protein we see on slide 3, that about 16 to 20% of an average adult's body is made up of proteins. This is the second most abundant substance in the body after water. We know that water is about 70% of our body weight, and then after that, 20% is proteins. So if you take all the water and all the fat out of it, the so-called dry body weight, 45% of that is made up of proteins. Where are these proteins? 45% of them are in muscles, 15% in blood, 15% in skin and connective tissue, and 25% in other organs. These are all aboutish. They're not exact, but kind of about percentages. So there are a ton of proteins in our body. And what do? So on slide 4, we see that for the most part, proteins do one of 4 things. They are either enzymes, meaning that they catalyze reactions in the body. Enzymes, their names all end in ACE, A-S-E. And so we have alcohol dehydrogenase, an enzyme that's in your liver that metabolizes alcohol. That's why when you drink a lot of alcohol, you end up with liver disease. Then DNA synthase that replicates the DNA, and RNA polymerase that translates DNA into RNA. There are many other types of other enzymes in the body. Those are just examples. They can be receptors. These receptors are on the surface of cells or the surface of nuclear envelopes. And what do? They bind to a signal molecule, to a molecule that is outside of the cell, comes and binds to them, and that starts a cascade of reactions that ends up doing what the cell is doing. So for example, olfactory receptors in our nasal cells, when a molecule attaches to them, they cause a signal inside the cell to produce a signal outside of the cell into the brain, and then we recognize that as the smell of, rose or fart or whatever. Or insulin receptors are on the surface of cells that they regulate how much sugar gets into the cell. There are a ton of other receptors. I mean, we were talking about in Dr. James's room, if you were there, we were talking about the SSRIs, the selective serotonin reuptake inhibitors. Those affect the serotonin receptors in the brain. We have dopamine receptors, norepinephrine receptors, cholinergic receptors. There are all kinds of receptors in the body, and they are all proteins. Another thing that proteins do in the body is that they transport things from here to there. So many things in the body are not water soluble. And so when they're not water soluble, you basically use a protein that is water soluble to kind of encase that and take it here and there. So, for example, hemoglobin is a transporter protein for oxygen, or high density lipoprotein transports cholesterol. Ferritin uh, transports and stores iron in the body. And so, if you really want to know whether you're anemic or not, you can't just go with your hemoglobin count or red blood cell count. You have to get your ferritin count too to see how much iron you have stored in the body. And the last thing, the last major thing that proteins do is that they're structural building blocks. So collagen, which makes our, skin, nail, etc. Keratin, the same thing. Actin and myosin are the two proteins involved in muscle action. So they're basically structural building blocks of the body, and their synthesis or metabolism is governed by steroids. And so the next chemistry series that I'm going to talk about steroids to see how they affect the metabolism of these things. So that's a preview of things to come. So on slide 5, I said that enzymes catalyze reactions. Well, what does that mean? What does a catalyst do? A catalyst is a compound that speeds up a reaction, but itself is not changed at the so the final form of a catalyst is the same as the initial form of the catalyst. So the catalyst goes through changes, but then always comes back to its original form, and that's why. But so it is not consumed in the reaction. So, for example, the reaction that I have written at the bottom, that's a base catalyzed hydrolysis of esters. So on the left-hand side, I have methyl acetate, which is H3C double bond O, OCH3, and this gets hydrolyzed with water. So as you can see, the water molecule on the right-hand side is in a rectangle, showing you that one hydrogen of the H2O went with the OCH3 to make HOCH3, which is methanol, and the OH of the water went with the H3C C double bond O to make acetic acid. So this is a hydrolysis. Hydro is water, lysis means breakup. And so hydrolysis means breaking up with water. And so we see here water comes in and splits the molecule into two, and the water molecule itself also gets split into H and OH. H goes with one, OH goes with the other. So on slide
But
six, yeah,
Sam, how
yes.
do we obtain the water out of this, or are you getting into it now?
Sorry,
Because
say that again.
the equation you put on the previous slide, it has
Uh-huh.
the water going in the compounds. Are you gonna get into the part where the water comes out again?
Oh, this is not catalytic.
Yeah. Okay.
So I'm going to now show you the catalytic way of doing this. So in the lab, this is slide 6. In the lab, we see how it is base catalyzed. Base here is OH minus, the hydroxide moiety. And so we see in the first step, hydroxide attacks the C double bond O, the carbonyl, to form that tetrahedral intermediate on the right. So the middle carbon is now attacked, it has 4 single bonds to 4 different substituents. Then we see that in step 2, the electron pair on the oxygen pushes out the OCH3 and forms a C double bond O. So now we have the acetic acid on the left and methoxy, which is methanol without a hydrogen, on the right. Methanol methoxy extracts the hydrogen from the acid, and then to make methanol, and then acid extracts a hydrogen from water to make acetic acid and OH minus. So now we see that OH minus starts out as OH minus in the reaction and gets out as OH minus in the reaction. So one OH minus goes in, one OH minus comes out. OH minus is not consumed in this reaction, and therefore it's a catalyst for this reaction. Did that answer your question, Shamash?
Yeah, I opened the mic too soon. I should have waited. Sorry.
No, it's okay. It's okay. So on slide 7, we see how an enzyme would do this. Okay, so we only have 2 of the amino acids in the enzyme. Now I do realize that a lot of the terminology I'm using here I haven't defined yet, I haven't talked about yet. And the good part is that I'm showing you this slide now, and this slide is also the last slide that I'm going to show. So we're going to go full circle. I'm going to introduce this to say this is what a protein does, and then go back and slowly uncover what this actually is. So just bear with me. We have 2 amino acids, histidine and serine. We have a molecule which is para-nitro phenoxy ester, and we have a water molecule. So we see that the nitrogen on the histidine extracts the H, makes an OH minus, that attacks the carbonyl group, but that is stabilized by the hydrogen on the serine. So that makes the C double bond O very much more reactive. And then the molecule rotates and it extracts a hydrogen from the histidine by getting also released from the serine. And so now we have on the one side PNPOH, which is that paranitrophenol thing, the ring with the double bonds in it, that with the OH, and HOC double bond R. So what happened is that the molecule that went into it got hydrolyzed. The bond between the O and the C was broken. And on the O part, we have one hydrogen, and on the C part, we have OH. So this is exactly the same reaction that I talked about, but is being catalyzed in a different way. And at the end, we get our histidine and our serine back in the original form that they went into the reaction. Enzymes catalyze hiding a different mechanism for the reaction than you would do in the lab. And they have all the components to do that reaction right there. And so that's how they can do it really fast. We will talk about another reason for why enzymes are so fast at these reactions. I invite people to kind of come up on the stage and talk about things. So it's not just a lecture, but a conversation, especially you, Jim. So let's go to the beginning of this story, which is, what is an amino acid? So everything I talked about so far was kind of, you can say it was introduction. And so now we're going to actually get into the detailed science of this in a way that hopefully will be. We have two classes of compounds, carboxylic acids and amines. A carboxylic acid is a compound that has that C double bond O OH group attached to it. One of the first things you learn in organic chemistry is R. R is any organic group that can be attached. So when you use R, it means that that's an organic group that doesn't get involved in the reaction, so we don't really care what it is. So C double bond OH is what determines what is something that makes that a carboxylic acid. So on the right, I have the R group, and the molecule on the right is a methyl group, a CH3. And so that is acetic acid, which is the acid part in vinegar. A second class of compounds we're going to talk about are amines. Amines come from ammonia. Ammonia is NH3. And so if you take one of those H's off and put an R there, so you have RNH2, that's an amine. And we're not going to go into what happens if you replace the other H's. Just this one, when one of the H's is replaced by an organic group. This is called a primary amine, and one of an example of this is methylamine. So I have NH2, which is the amine part, and then CH3, which is the methyl part. So as you can see on the carboxylic acid, let's say on the acetic acid side, on the left, I have a CH3. On the methylamine on the right, I have a CH3. So those two CH3s can basically get combined into one and attach the two molecules together. So As I have shown on slide 9, the amine H2NR and the carboxylic acid are COOH. I can delete one of the R's and have the other R bridge the two. So I would have H2N, that was the amine that came from the, that's the, yeah, amine that came from the amine molecule, and then R, whatever it is in the middle, and then COOH, the acid of the carboxylic acid. Now, if that R group in the middle is a single carbon that has a hydrogen and another R group attached to it, then we have what's called an amino acid. So an amino acid is a molecule that on one side has an amine group, an NH2, on the other side has an acid group, a COOH, and in the middle has a single carbon with a hydrogen and another moiety attached to it. Any molecule that has this format is an amino acid. So on slide 10, we talk about the different uh, parts of an amino acid molecule, because all of these parts become really important when we talk about proteins. Usually we write protein sequences with the nitrogen, the amine side on the left, and the acid side on the right. And so this is that is the direction of the protein sequence that's conventional is N to C. And so that's why all the amino acids that I show on these slides, they are in that format, that N is on the left, C is on the right. So on the left-hand side, inside the red box, that's the amino end, or the amino terminus, or the N terminus. All of these terms are used, and all of them are used extensively. On the right-hand side, in the blue box, we have the acid end, or the carboxylic terminus, or the C terminus. So this is why, because of the N terminus and the C terminus, a minute ago when I said the way we write the directions from N to C, it's from N terminus to C terminus is the direction of the amino acid chain by convention. The single carbon that is in the middle is called the alpha carbon. Alpha comes from the old naming terminology for carboxylic acids, where the first carbon after the carbon of the acid was alpha, and then beta, da 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 da. And so for those of you who know what GABA is, it's is gamma amino butyric acid. So I know it's an acid. Butyric has 4 carbons, and it's gamma. So I go alpha, beta, gamma. So it's the third carbon past the acid carbon. So anyway, this is an alpha carbon. It has a hydrogen, and in the black box, it has a side chain. So what determines what amino acid you have is the identity of that side chain. Now, if you know organic chemistry, or if you were around in some of my past lectures when I was talking about the tetrahedral structure and chirality, the handedness of a molecule. So we said that if you have a carbon that's attached to 4 different substituents that are not identical, then you have a chiral center, meaning that you can have 2 different orientations of the R and the H, where the mirror image, they're not superimposable. And so that's a chiral center. So on slide 11, I have shown the kind of the way we draw 3-dimensional structures on a 2-dimensional surface. The solid wedge is coming out of the page, and the hashed wedge is going into the page. So on the NH2, we have a hydrogen coming out of the page, a hydrogen going into the page. And on the alpha carbon, we have a hydrogen coming out of the page, an R group going into the page. If you were to draw the mirror image of this, you would find out that the mirror image of this molecule is not superimposable on this molecule. So this conformation, as I have drawn it in biology, is called L amino acid. We chemists don't like the L, and so the mirror image of it would be D. We don't like L and D designation. Biologists do, because we being chemists, because if you say L, I won't be able to draw which isomer I have. So we use the nomenclature of R and S, and with R and S, it has basically a method of determining whether something's R or S based on the arrangement of the substituents. And so if I know if it's R and S, then I can draw the molecule. But L is the isomer that interacts with biological uh, uh, things, whereas D is the isomer that doesn't interact with biological things. And so this is what biologists like the L and D. They don't really care what the structure is, they care whether it's going to interact or not. So all the amino acids that are used in our bodies, actually all amino acids in nature, total, in plants, amoeba, animals, whatever, all amino acids that are inside a living being are L amino acids. We do use D amino acids mainly in the lab, not in biology. So on slide 12, I have the listing of the 20 common amino acids. Why is it that 20 common amino acids? Because these amino acids are the ones that are used in proteins in nature. So again, from amoeba all the way to us and plants and bacteria and everything, all of our proteins are made out of these and only these 20 amino acids. Why is it called common? Because we have many other synthetic amino acids, but they're not as common. So in this chart that I'm showing you, it shows what the side chain is. So we see that glycine doesn't have a side chain, or its side chain is hydrogen. And so because in glycine, the alpha carbon is not attached to 4 different things. It's attached to 3 different. So that one is not chiral. L or D is just glycine, but for the rest of them, we do. And so the side chains are in red, part is in black, so you can Or actually, they're in color. The color is based on the functionality or the grouping of the amino acids. You see their names are in bold, but we basically in naming them, we either use a 3-letter code or a 1-letter code. So for alanine, the 3-letter code is ALA, and the 1-letter code is A. Valine is Val, and V all the way down. So when you see protein sequences, they're either in the 3-letter code or in the 1-letter code. You never see the full names spelled out. Slide 13 is just the same thing in a table format. It's a very good reference thing. And so those are that ends the talk about amino acids, and now I'm going to go into peptides. Are there any questions, any comments, anything on our discussion of amino acids? Because these things kind of tend to build on each other. If not everyone all at once, one by one. Jim, do you have anything to add? You don't. All right. So we go to slide 14. We talked about combining the amino and the acid from amino being left, acid being right, and then, putting them together to make the amino acid. Now, what happens when you have two amino acids, right? One has a carboxylic acid on the right, and the other one has an amino group on the left. Well, these two can react and form an amide. Okay, this is called a condensation reaction. What happens during condensation? Water gets out of things, right? Water gets condensed. It comes out of the air and condenses, on your glass or whatever. And so in this condensation reaction, what happens is that we lose water. This is exactly the opposite of the hydrolysis reaction. So we saw in a hydrolysis reaction, water comes in and breaks up the molecule. Here, the two molecules come together and they lose a water molecule and they combine. So I have kind of put in the box the OH and the H that are going to combine to form the water, and the C of the C double bond O and the N of the remaining NH are going to bond together to form a C double bond O NH middle group. That is an amide bond. So an amine plus an acid forms an amide. If this amide bond is between two amino acids, meaning the amino of one amino acid and the acid of another amino acid, then this amide bond is called a peptide bond. So a peptide bond is a special case of an amide bond where the bond is between two amino acids. This is where we get the word peptide from. And so on slide 16, I have shown in more detail, more schematic detail, how a peptide bond forms. So this is kind of color coded. You guys can see what things are coming off and what things are getting attached. So that peptide, don't ask me what the etymology of the word peptide is. I don't know, and if anybody does, I'd be more than happy to hear that. So what does peptide refer to? It refers to both the bond, it's in a peptide bond, and the product. Like I have a peptide molecule. So it's both a peptide molecule and a peptide bond. A peptide chain is a polymer of amino acids. How long can this chain be? Infinite, right? It could be 2 to infinitely many. It's all a peptide chain. Does a peptide chain need to have any biological function? No. All it is a chain, a polymer of amino acids. But the peptide chain can have 3 different specific names. One is an oligopeptide. An oligopeptide is a peptide chain having a small number of amino acids. Oligo is I think it's a small number, meaning like oligarchy, right? And so how small is small? Well, it's up to you how small you want it to be. I'll talk about that a little more in a little bit. Polypeptide, poly means many, is a peptide chain that has a large number of amino acids. What is large number? That's quite subjective. Depends on the context that you're talking about. What is a protein? A protein is a large polypeptide that has a biological function. What is that biological function? Well, that's also subjective. So basically, it's like definitely something that has 3 amino acids in it. Most people call that an oligopeptide, not a polypeptide. Something that has about 100 amino acids in it, people call that a polypeptide, not an oligopeptide. Something that has 300 or 400 amino acids to it, that's mainly a protein, not a polypeptide. But where is that transition? It depends. And so, Jim, correct me if I'm wrong, but I don't think anybody is going to take umbrage with you if you call something a polypeptide or an oligopeptide. It's basically up to you. What do you think, Jim? Am
Lost you
I?
there. Can you hear me?
Yeah, yeah.
Can you hear me? Yeah. No, you're right. It's inexact terminology. Nobody's gonna get their pennies in a wad there. Hey, I need to jump off, but before I do, um, I gotta get on my soapbox here for just 1 second. I'd love to the structure part of it that you're about to get into is my favorite part, and I'm really bummed that I have to miss it. But one of the things that you haven't really talked very much about is the role that proteins play.
Jim, you're in the matrix. We have a hard time hearing you.
Okay,
You're
you know what?
breaking
I'll
up.
follow up with you. I'll follow
Okay,
up with you. Great lecture.
thanks. Yeah, sorry about. I mean, Jim is the molecular biologist, and so it's too bad. He's driving and he's kind of going through dead zones. Uh, anyway, so that was the terminology slide. I have another terminology slide on slide 18. So a monopeptide is a single amino acid. A dipeptide is 2 amino acids join. Tripeptide is 3 amino acids. After that, we kind of dispense with the Greek, and we just use this mer terminology. So 4 amino acids is 4 mer. The mer comes from polymer. So the poly part of it, you basically assign a number to, like 4, 5, 6, 785, whatever. And so you just call it a 6 mer, a 7 mer, 785 mer, because what 785 is in Greek, except for the Greeks. So that's how we refer to polypeptides. So we described really what an amino acid is and what a peptide is. And up until now, if there's any biological function, it's mainly as part of signal transduction. It's regulation. It's kind of like it binding to DNA as polypeptides to stop the transcription of DNA or the replication of DNA, things like that. And so they, as of now, the molecules we've been talking about do not show any catalytic function. So we go to slide 19. This is the start of the protein discussion. The first thing that we talk about about proteins is their structure. What do they look like? And the structure of a protein, we have 4 different structural designations: primary, secondary, tertiary, and quaternary. The primary structure is the sequence of amino acids. And the sequence of amino acids doesn't have any structural component to it. It's just the long sequence of the amino acids. And something like this doesn't really exist in nature, because as soon as the chain is being synthesized, the components kind of organize themselves in what is a secondary structure. So on slide 20, this is the local arrangement of amino acids in the chain. And we have 2 different arrangements in our secondary structure: an alpha helix or a beta sheet. Slide 21 is the alpha helix. So we see on the left-hand side that the amino acids kind of arrange themselves in a helical structure. It's a right-handed helix, like all other helical things in nature. And there's some data about the length, the degrees, the things that we don't really go into. But what I want to show you is on the right-hand side, you see that hydrogen bonds form between the hydrogen atom of an amino group of one residue to the oxygen of a carboxyl group of another residue. And it is this hydrogen bonding that kind of pushes the amino acids into this helical conformation. But this is local, right? So meaning that this helix will go for, it says down here that the typical length is about 10 amino acids. It kind of dies after the 10th amino acid. The other local conformation that we get on slide 22 is the beta sheet or beta pleated sheet. It's both terminology is used. They're the same. This is when there's hydrogen bonding with not the amino acids in the same section of the chain, but you're forming hydrogen bondings with amino acids in a different part of the chain. So you get this going back and forth pleats that you have hydrogen bondings between the lines that go back and forth, and you see that on the left bottom picture, we start from top left and we go and we get to the two red amino acids. The red two amino acids is called the beta term, and that's when the amino acid sequence kind of folds onto each other and goes back along the same path next to the sequence that it came from and forms hydrogen bondings with that. And the hydrogen bondings are also shown in the right side picture. So those two kind of are the secondary structures, the local conformation of the amino acids. After that, the whole protein kind of folds onto itself and forms a shape that if you kind of look at it from the outside, if you don't go down into the molecular level, it looks globular. It looks kind of like a spheroid object. But when you go down in there, you see that no, it actually is made up of a lot of different conformations. So on the on slide 23, we're going to talk about the tertiary structure of the protein. That's when the whole thing folds over itself to kind of form the final protein that has a function. And we see on the left-hand side, we have the primary structure, which is the sequence of the amino acids. A1, A2 stand for amino acid. And then we get to the middle part, and we're forming the secondary structures, the local conformations, alpha helix, beta sheet. And then on the right-hand side, we have the tertiary structure. So now you see that the amino acid sequence kind of goes around. Sometimes it's in an alpha helix, sometimes it's in a beta sheet as determined by the funky arrows, and sometimes it doesn't have any secondary conformation. It's just floating about. Those are the thin lines as opposed to the thick purple lines. So how much of an alpha helix are you going to have? How much beta sheet? How much free floating? All of this really has to do with the primary sequence of the amino acid. It's the primary sequence that basically determines how things are going to fold onto each other, because the primary sequence is one that determines where hydrogen bonding can occur. So a lot of proteins, their structure ends right here. They're basically what we call single-stranded proteins, meaning that there's or single chain or single subunit, which doesn't really make sense. So it's a single chain protein, and it forms. So myoglobin. What is myoglobin? Myoglobin a protein inside our muscles that has a heme group, the same as a hemoglobin, has an iron in it, and this is what transports oxygen from the outside of the cell into inside of the cell. So it's an oxygen transport molecule inside of our muscle cells. Why do we need that? Because muscle cells use a lot of oxygen, and so they need to have this other protein to bring oxygen to the inside of the cell, distribute the oxygen inside of the cell really fast. And so this is why muscles are red, right? Red meat. It's red because of the myoglobin inside of it. And so chicken breast that doesn't use aerobic metabolism, it's completely anaerobic metabolism, doesn't have any myoglobin because it doesn't need to transport oxygen. So it's white, it's not red. But chicken legs, they have myoglobin, so they end up being red. Anyway, so myoglobin is a single chain protein, but we're going to go now on slide 24 talk about quaternary structure. What is quaternary? This is when two or more of those tertiary structures come together and they aggregate and they form a protein. So hemoglobin, not myoglobin. Myoglobin was oxygen transport inside of muscle cells. Hemoglobin is oxygen transport in the blood. Hemoglobin is made up of 4 different proteins. We have 2 alpha chains and 2 beta chains. And these are folded differently, and they all come together to form the hemoglobin protein. So because all of these chains, they now are called subunits. Every one of these subunits is a complete protein with a heme in it. So hemoglobin ends up having 4 heme molecules inside of it. So each hemoglobin molecule can carry 4 oxygen molecules instead of myoglobin only carries 1 oxygen molecule. So the quaternary structure of the protein is the aggregation of these subunits. So we had primary structure was the sequence of amino acids. Secondary structure was local conformation of the amino acids into alpha helix or beta sheet. Tertiary structure was the folding of the entire chain onto itself to form a protein. And quaternary structure is 2 or more of those folded proteins aggregating together to form a protein. Many proteins in our body are quaternary, meaning that they are made up of 2 or more subunits. Any questions so far?
No, not yet. Thank you.
All right. Okay, so how do these proteins then do the catalysis that we were talking about? The first thing is the concept of an active site in slide 25. An active site is kind of a cave or a hole that is formed in the structure of the protein. that another molecule can go and inside of it and fit in there and then get subject to chemical reactions inside of that cave. So I have shown you here a
Wait,
protein.
Sam.
Yes, slide 25.
So I'm really late. Okay, I do have questions, but I'm so far back that let me read and try to
All right.
catch up.
Yeah, yeah. Let's we can talk about that at the end.
Okay.
All right. So we see this protein. We're looking at all of the alpha helices. We see all the data sheets, and we see that a molecule is inside of that active site. So what happens inside of that active site? This is what I promised at the beginning. So now we can understand this slide 26. We see on the left, we have a histidine. We now know histidine is one of the amino acids. And it has a number next to it, 265. So this means that histidine is the 265th residue in the primary structure in the sequence of the amino acids. To the right, we have serine, 112. So serine is the 112th amino acid. And so you see that the protein has folded itself in a way that residue 112 is right next to residue 265. And so our ester molecule can slide in and fit right between that histidine and that serine. This molecule is now inside the active site of the protein. And now the protein can do the amino acids. What do? They're extracting hydrogen, they're adding hydrogen, right? And so they do all of this to make the electronics of the molecule for the reaction that's going to happen, which is the attack of water to that carbonyl compound. This makes that electronics a lot more favorable, and the orientation of the molecules are such that they're right there. They are exactly in the place they need to be for the best orientation of this reaction. So you don't have to wait for a whole bunch of attacks until finally one comes in at the right orientation, which is what normally happens in a chemical reaction, right? So if we were to do a statistical mechanical analysis on this to see how many fruitful collisions we're going to have here, it's going to be 100%. Whereas in a regular reaction in a round bottom flask, it's much less than that. And so then we now see that at the end, histidine 265 went its own merry way, the same way that it was at the beginning. And so does serine 112. So we basically, I think, giving the talk, we can understand this reaction of a protein. How is it that the protein primary sequence leads to this tertiary structure that creates this active site? that now this active site can create the right conditions for the reaction to take place, and the reaction is enzymatic. So one last question remains for me to answer, and that's well, why do enzymes work so fast, right? The thing is that almost all reactions in our body are mediated by enzymes. Why is that? Because if we wait for the reaction to go the way it does in a round bottom flask, that's way too slow, right? Those reactions, you know, they may take 5 minutes or 10 minutes to go to completion. And by then you're dead. If you have to wait for a reaction in the cell, if you have to wait for 5 minutes, you're dead. So why do enzymes work so fast? Because of a concept in chemistry that we have, that we call the neighboring group effect. So if you have the two reactive moieties locked in position next to each other, then that is called a neighboring group effect. That is the two reactants are neighbors of each other. The local concentration of a neighbor is huge. Okay, why? Because it is right there. It's when the one of the reactants, you know, looks up, sees the other reactant right in its face. Doesn't have to wait for it to come around on the guitar for it to see it again. It's right there. So we look at the rate equation for the reaction of enzyme plus substrate goes to products. We see that the rate is rate constant times the concentration of the enzyme to the power of the order of that. We can kind of forget about what the order is at this point. It's not critical here. Times the concentration of the substrate to the order of that. Well, the concentration of the enzyme in the cytoplasm is 10 to the minus 6 molar. That is, it's in the micromolar, and for a lot of them in the nanomolar, 10 to the minus 9 molar concentration. Molar means moles per liter. A mole is Avogadro's number of molecules. So 6.024 times to the 10 to the 24. And so that's how we measure concentrations as number of molecules per volume. So here, moles per liter. So the concentration of enzymes is very, 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 very low, right? Micromolar, nanomolar. But but in a neighboring group inside of the active site, the concentration of those components are somewhere around 10 to the 8 molar. That means you have 14 orders of magnitude greater effective concentration of the reactants inside an enzyme's active site. And so if you take that E inside of the brackets from 10 minus 6 and make it 10 to the 8, now you can see that the rate of the reaction basically jumps up by 14 orders of magnitude. And so this is in a very, very simplistic way, right? As a kind of as a first approximation. This is why enzymes work very fast. Well, this concludes what I wanted to say today about proteins, enzymes, etc. And thank you very much for being here. Please come up and let's have a conversation about what we just talked about, and also for my throat to rest.
I came into this late. Is this an ongoing lecture series?
So in the Scientific Discourse house, every Friday at 9:30 Pacific, 12:30 Eastern, we have a room, and there are three of us as kind of leads in the house. Each one of us kind of we kind of like at the beginning of the week we kind of say, well, who's going to do it this week? And sometimes we ask other people to join us. For example, Hiro, Shamash, and other people. So yeah, we have a scientific room every Friday. The rooms that I do are all life sciences, chemistry, and biochemistry.
Nice, that's amazing. Do you ever watch TWIV?
What?
This Week in Virology?
No.
Do you? Back during the pandemic, they had a whole like course on viruses that was like the same course as the university, but they just did it on YouTube and anyone could participate.
Uh-huh.
So sort of similar to this, but like maybe a little bit this teaching materials were a little more formal because they were specifically from a Columbia University class, but they just including me.
Yeah, well, these rooms are designed to be informal. First of all, I don't get paid to do this. And so we're just putting together some slides to talk about topics we know about. And they're designed to be, for non-scientists, but people complain that that I I have a hard time bringing the level to be understandable by most people, so I apologize for that.
I thought the thing about myoglobin I found fascinating, but I got here kind of on the tail end of it. Do you know what ELI5 is?
The what?
ELI5, it was a Reddit phenomenon that stood for Explain It Like I'm Five.
Oh, no.
It basically means to break things down as
Yeah,
like the simplest of terms. If
yeah.
you search ELI5 in Reddit, you can sort of check that out. It's definitely a
Well, the thing
skill.
is, the problem with like a talk like today's is that if I want to do that, then this talk would be, 14 weeks long. I have to assume that basic organic chemistry. I have to assume that when I say oxygen attacks the carbonyl, what I'm saying, right? Because I have talked about those in previous talks, and so I'm kind of assuming uh, that I can't repeat all the talks every single time. So
For
so
sure.
at some point I have to say I'm going to assume this basic amount of knowledge, and then go from there. That's the only thing I can do. Hi, Ben. Welcome.
Hey man, sorry, I'm here with people, friends. They just mentioned peptides, so thought I'd see if your room's still going. But we don't have any questions about them right now. But I'll read your slideshow, man. Thanks for doing the room.
Sure. Well, the formal part of it is over. We're in the Q&A part.
Well, hey,
So,
what's up, guys?
Hi.
hey, so Sam, that was really good. Thank you for that. The whole talk. I feel like that's good background info that I didn't really have, or not much of, anyways. But do you have any knowledge or any info about the history of peptides as medications? Because I know, GLP-1 type stuff are in the news, and also all these peptide things that are being hawked are out there in the whatever biohacking manosphere world. And it's been sort of infringing on my world like quite a bit lately. So I don't know if this is like a completely new thing or if this is something that's been being used for a long time, and these are just the latest examples of them. But do you have anything to say about that?
Yeah, so the use of biologics, first of all, most of the biologic uh, medications are antibodies. So basically, antibodies are proteins that recognize a portion of the surface of another protein and bind to it. And then that allows for uh, the immune system to recognize that protein as a foreign antigen and destroy it. So that protein can be like a cell surface protein, a receptor protein, and so then the immune system would recognize the entire cell to which the antibody is attached as a foreign antigen and destroys it. So most of the medications out there are now in the form of antibodies. So antibodies have been used for a long time. Monoclonal ones were developed in the late 70s, but they were not used as medications, mostly because we didn't have the technology to kind of get them into the body. That was very difficult to do. Every time we were trying to physiologically apply them, they would degrade. And so that problem wasn't solved until the late 90s, mid to late 90s, how to get biologics into the body. And we have figured out how to get protein biologics into the body without much difficulty nowadays. We're still we still can't get DNA and RNA into the cells of the body. We can get them into the bloodstream, but we can get them be taken up by the cells. So I would say the history of biologics as medications goes back maybe 30 years.
Well, Sam, I got a question about. I was considering to be a part of this study out here in Florida. It's called the lipoprotein study. Um, so what they were talking about is that there are some people who have, uh, who are like born with, these high blood cells. Well, I mean these cells that cause high blood pressure, and like it's you might know what I'm talking about, but it's called the lipoprotein, and it's basically an extra layer of fat that is attached onto the blood cells. So whenever the person were to like eat a nutrition something nutritional, they'll get um they'll get high blood pressure, and it can essentially cause a lot of health problems. So I was just trying to like because they was really looking to like study people that have it, but like I was just trying to understand it because they was like they have a study going on about it, and I know my dad has like high blood pressure, so I was just trying to consider if you
Yeah,
know.
I don't know anything about specific clinical trials. Sorry. And I'm not aware that lipoproteins are involved in blood pressure regulation. They are involved in cholesterol transport. So if you have high triglycerides or high cholesterol level or uh, that, but maybe if the high blood pressure is because of a lot of cholesterol in your blood vessels, in your endothelia, then that might be it. But I don't know. I don't know
Yeah, that was it.
the
Yeah, that
details
was
of
it.
yeah.
Yeah, that was it. That was it. It was the cholesterol. So what it was like these extra clumps of cholesterol like in your blood vessel is what causes all of that. So that's what they were
Yeah.
yeah.
All right. So this room was based on a recommendation from Otter in the audience. So I want to know if Otter, did this meet your satisfaction, or do we still have questions that are unanswered?
Yeah,
Uh,
what's a
I would like to
peptides?
know. I recommend you go back and listen to the replays after the room is done. I'm not going to repeat the room for you.
Yeah, I may have a question, Sam.
Uh-huh.
That for the for the four the main roles like enzymes, receptors, transporters, and structural building blocks. If you had to rank these, maybe by the main like emergent physiology, like how an organism maybe behaves as a whole. Which category you think sort of dominates maybe?
Yeah, they're listed in the order of dominance.
Oh, okay. I have to go
Yeah.
back myself. I came in late as well.
Yeah. Oh, I don't think I said that in the talk, but so actually that was a good question. I probably should have said that. These are listed in terms of order of importance, or I can't say order of importance. I mean, if you don't have any of them, you die. They're all important, but in terms of the number of things that they do, the prevalence of their function, or the prevalence of the proteins, this is the order.
Oh, okay. That's yeah, I'm going back. Oh, okay. Okay. So what about so if you think of a receptor as a specialized like information interface, um, how it's not just a binding site, that right that
Sorry. Can you start over again? I got distracted a bit.
If you're thinking of a receptor as a like a sort of specialized information interface, um, rather than just a binding site, um, can that change the way we might conceptualize this?
It's not just a binding site, right? If it were just a binding site, then it wouldn't do anything. What it does is that when the signal molecule, which is outside of the cell, binds to, so the receptor has three portions to it. One part of it is outside of the cell. One part of it is inside of the cell wall, and then one part of it is inside of the cell. It has all those three parts, right? So, and this is why it can connect the outside of the cell to the inside of the cell. So when a signal molecule binds to the outside part of the receptor, that causes a conformational change, meaning that the tertiary structure of of the protein changes the way it is folded, because now you have added something to it. The structure of the protein changes to accommodate the binding of this receptor, and that conformational change opens up the active site inside of the cell. And so then that active site can go and do a catalytic reaction on another molecule, which is now the start of the cytoplasmic signal transduction. So it's the receptor is like a switch that you put the key in and you turn, and it connects the electrical signal inside of the cell.
Oh, thanks for that. Yeah, I thought I had another question. Well, I don't know if anyone else has a question right now. Thanks for explaining that more.
Sure. So, Kyle, what's the difference between signals and noise when cells metabolize? I'm not really sure what you mean by that question, but the thing is that a lot of receptors have basal function, meaning that without having any signal molecules attaching to them, they do catalytic activity. And what the signal is, it either makes that catalytic activity more, where we call that an agonist, or they make the catalytic activity less, and we would call that an antagonist. And so in the studies that you do to figure out the potency of your agonist or antagonist, the noise is the basal function of the receptor. The signal is the function of the receptor with that agonist or antagonist. So I'm not sure if I understood you fully, but that's what I understood.
Well, that made me think of question. So, what's the most like interesting open questions about the how the transporter proteins modulate the systemic risk, like cardiovascular or metabolic, that we don't yet quite know, maybe
All of them,
all the above. Okay.
yeah, because every signal pathway, when it doesn't work optimally, causes a disease, right? A disease we've talked about is a non-optimal function of the signal transduction pathway. So any of these pathways, when they don't work optimally, they cause a disease. And so they're all important. Some of them, because of their prevalence, are subject of greater research. So, for example, there's like the C28, 24. Those are CD28, CD24. Those are some of the pathways in in cancer, or I mean, you have the types of breast cancer. If you have, HER plus, HER double plus, HER triple plus, right? Those are all different signaling pathways that cause different types of breast cancer. So they're all those are, because of the prevalence of those diseases, those are the ones that are studied more, but they're all significantly important.
Gotcha. Yeah. Thanks. I'll go over. I'll listen to what I missed.
Very good. Usually when there aren't many questions, it's either that the subject matter was uh, very clear and no questions remain, or nobody understood what I was talking about. And I think because of the
Well,
way
I
I
just came
presented
in.
this, this falls into the fact that it's like, what the fuck did you just say? So,
Well, I'm trying to figure out how I always do when I'm learning something. I try to figure out how to relate it to my life. I have a
okay.
daughter. I have a daughter who has just been diagnosed with breast cancer, and I see that I missed the part where you talked about the peptides and the proteins, but she's been, looking into peptides and proteins for herself. So any any you don't have to do it online here, but any advice I would appreciate.
No, I wouldn't dare uh, on a situation like this to when someone has a significant ailment to say anything that doesn't come from the oncologist. I mean, I'm of the opinion that in these situations, your oncologist should be your only source of information. But very generally speaking, there are a lot of antibody treatments for cancer that they basically cause the body immune system to attack cancer cells.
Yeah, and
And
that's what we're looking for in addition to what she gets from her four
right.
doctors.
Uh, uh, uh,
Never
no,
mind.
yeah,
I'll try
I
to
don't, I
I'll
don't want
try
to
to
say
Well,
get through
anything here.
I'll
Sorry,
try to
it's
get through your slide presentation and
yeah.
see what I remember from the Bronx High School of Science.
Yeah, it's something like that. I really don't want to say anything that because you're dealing with a serious illness, and you have to go by the experts, and I'm not an expert oncologist.
Right,
well,
I
Dr.
understand.
Francine's question taps into my motivation for this conversation, which is to talk about this kind of larger social thing that's going on right now with these kind of gray market peptides and um,
Exactly.
and um, I do want to have that conversation, but I should have predicted, Sam, that you're the kind of person who's smart enough to not go into like, yeah, this is all really really good background, but frankly, my motivation is to have the conversation that Dr. Francine wants to have too. I think, but
So so I I'm okay having that conversation, but I'm not prepared to have that conversation. If you allow me to put together another scientific discourse room on peptidic medications, then I would be in a position to kind of have fresh information to talk about it. If I want to talk about it now, it's basically going to be off the top of my head, and who knows if I remember shit correctly or not. So I have to be very careful.
Yeah, there's just so much noise in the system about peptides and proteins and, what they're used for. I appreciate you're not wanting to talk about that, without being prepared, but you should get on Reddit and see how unprepared everyone else is.
Oh yeah,
Well, that
I I
yes,
am fully
And they're
aware of
wildly giving
that.
advice and selling things.
Yes.
I would
that
love
is the big
to.
problem, right? Like the people who know better won't talk about it, and the people who don't know are very willing to talk about it and very willing to sell it and give advice and copy other people's websites and all this shit's going on.
Exactly.
So it's a Um, so and I've only just kind of become aware of this like in the last couple of months, but I'm late to the game. This thing has been a thing for quite a while, and RFK is up there in congressional hearings saying that peptides should not be regulated by the FDA, and that people should just be free to explore these things on their own. That seems like just
Uh,
nut job craziness to
you
me, but
know, it's very difficult with RFK to kind of say, well, that's the dumbest thing I've ever heard, because that kind of goes for everything he says. So it's like,
Yeah.
how do you determine which one was the dumbest one? That's very difficult to do.
Sometimes South Park had an entire episode on this and lowering the bar, and it's kind of like they, as soon as they get the bar at ground level, they get subterranean, then they have to go to the ocean, and yeah, it's just some of the stuff. I don't know if you heard about the flu vaccines in our military and what went on with that,
Yes.
but just it's just wild. I was
Ne-
Yeah,
going to
they're
ask
going
him if
back to it, right? Because
yep,
it's
yep,
like,
one man
guys,
died,
this
14
shit actually
hospitalized,
works.
200 to 300 cases. One soldier appears to have died. 20 others hospitalized, some in critical. Yeah, it's we forget that the flu is not a joke. But I was going to say,
No, we
Sam,
forget that science works.
on
That's
that too. On a related note, if I would love it if you did do, I'm kind of new as well to the whole, kind of like here, I'd say I'm aware of peptides, but just like in a chemistry course, like what you taught earlier, I learned that, of course, back in the day. But this whole like hyper use and treating of everything, and obviously the GLP-1s and stuff like that, peptides are having their moment in the sun. I'd be very curious to hear about the treatments, the legitimate and the iffy. I'm also very curious because I think you're probably uniquely qualified to talk about maybe the safety and process, because when I was hearing they were unregulated, it sounded wild. I know that there's a huge problem of regulation with supplements where they're literally unregulated and often don't even contain the compounds they claim they do. And it's wild to me that peptides that we inject might be in that same category. So I'd be really curious to hear like what are the standards around these labs? It certainly seems like people aren't dying left and right, so there must be, but I'm just quite curious. And also I having heard the news out of China and what they're doing at some schools with administering them to entire student bodies while they're taking standardized testing is wild. So it's US, it's China, I'm sure it's elsewhere in the world. So I'd be very curious, if you can talk about addressing kind of those angles as well, if you're able to.
uh very generally, uh, uh basically, if anything, that this is my own personal thing. Okay. Uh, on uh on illnesses that are not really illnesses, right? Like you get the cold, or you get a headache, shit like that. I personally, I don't care what people do, right? You wanna boil, I don't know, uh, poison ivy and drink that to cure your headache. Go ahead. I don't care.
or put it on your face.
Yeah,
I mean,
poison
Sam,
ivy
who
cream.
who doesn't love a good mercury tincture, right?
I know. So, but when it comes to diseases that are real diseases, right, meaning that they can actually kill you, or they can make life really miserable for you, right? They're long term, they're this, they're that. A kind of disease that you would go to the doctor to. right? Usually colds and flus and things like that, we don't go to the doctor. It just passes, and we just do home remedies of, you know, orange juice and kiwis and shit like that. So for an ailment that you go to the doctor for, personally, I mean, this is, I'm not telling you what to do. I'm just telling you what I do. If the medication doesn't have a package in an FDA issued package insert, I do not take it. That to me is the standard that the manufacturing was done correctly, the controls were done
That's a
correctly, all
pretty
the
good
tests were done
standard.
correctly. Yeah.
That's a pretty good standard. It's not perfect, but it's a pretty good one.
And because it also doesn't tell you that it works, it tells you the whole pharmacology of it, the whole biochemistry of it, all of that with references, with this, with that. So if you really want to, you can go dig really deep into the package insert and figure out what it does. So and the thing, I mean, manufacturing of these things, there are processes. One's called GMP, the general manufacturing, the good manufacturing protocols. Another one is CGMP, the clean good manufacturing protocols. These are different protocols in a lab in terms of cleanliness, in terms of how reagents are handled, how instrumentation is handled, what kind of clothing the lab personnel wear when they do all this stuff. So almost all things that go into our bodies require a CGMP manufacturing protocol. And if you do not have CGMP, the FDA won't even look at your application. And
Are
after
these
you
peptides?
start, after you start, the FDA visits you and from time to time to make sure that you do have those, you are following those. And
Like a
even,
restaurant inspection.
yes, exactly.
But just
And
to double
even
check,
if, yeah, yeah,
Sam, are those peptide labs? This is kind of the question I have. Are they a lot of supplements? Some are manufactured with this kind of standard, right?
right.
Um, but some are not, and some are manufactured with functionally no standard. I mean, they just did a thing with like Timu importing samples, and one of them was like I think it was a lead tainted. I can't remember what they claimed it was. It ended up being just like soy powder, and it was tainted with lead and chemical dyes.
Yeah.
Um,
So
so I'm
so
curious
those
how,
are,
are the peptides regular? Like, if somebody goes for a peptide treatment at these biohacker facilities, are those all coming from an FDA certified lab,
No,
or might
absolutely
those
not.
be coming?
depends.
They're not, well,
Yeah.
the sites that are popular and are being talked about, Sam. I think if you learn about these, it'll make you very, very angry because I think none of this stuff is going on.
No, I
Yeah,
know
yeah.
that
Okay.
because I mean the thing is that they have to somehow get approval from the FDA to sell this, and the way they do it, they get approval as I mean they say, oh, this peptide comes from a plant, so it's a plant extract, and so we don't need to do any of the clinical trials, right? Because you don't need to do that for, I mean, if you had to do that for plant extracts, you had to do it for your tea and for your coffee. But so plant extracts don't go through an FDA approval process. So if they're saying that their peptide is a plant extract, or if their peptide is, I mean, there are whole categories of things that you put into your body that don't come under purview of the FDA. And so if they are marketing their peptide as one of those, then it doesn't see the FDA, right? And
That
so,
that's my I'd be interested. And last quick question, Sam. This is a deep cut from earlier. I'm very interested in chirality for a number of reasons. Back in the day, it was always an interest to me. You mentioned that in the lab, you typically use D versus L chirality. Is that just to eliminate potential bioreactivity,
Uh,
or is there another reason why they tend to use D in the lab, or like it's left over from reactions?
oh,
Like,
the
what's the reason for that?
DNL are leftover are used in biology because they're leftover from the time when we uh, couldn't do structure elucidation. So we didn't know what the structure of the compounds were, but we knew that these ones had biological functions, those didn't, and so these were L, those were D. It
In other words,
comes.
you mentioned like, do you intentionally try to use D-chirality of a lot of these compounds in the lab um, to avoid bioreactivity? That was just, I was trying to think why you would intentionally use D, or is it just like to make sure all the reactions go through? D was just picked as the standard.
So L is what almost everybody used. D is just used for research purposes. So there are a lot of things that you want to do, uh, products that have like a certain property, but they don't interact, right? So in the late 80s, we had this oil that was D instead of L. And so that oil, right, it wouldn't get absorbed by the body because it was D. And everybody was so excited about it. But then what happened was that, well, it was still oil, so it wouldn't get absorbed. So what happens to an oil that doesn't get absorbed? It causes violent diarrhea. So that was a no-go. So there are a lot of things that are made that way, that you want to have a a, a let's say proteinaceous solution because of the density gradient that it creates, but you want it to be biologically inert. So you make those proteins out of D amino acids.
So Breyer set me up, gave a perfect segue to the question I wanted to ask, which is even more speculative than peptides being sold on in the black market right now. The whole issue of the homochirality of known life on Earth, and how it is that maybe just by random selection very early on, the L chirality was settled upon for life here, and I wanted to bring up the ban that's been proposed, I think is enacted, on doing research on creating quote unquote mirror life from our chirality.
So, sorry, your specific question is?
Um, I just wanted to hear your thoughts about this whole notion of the quote unquote mirror image life, and the likelihood that anything that happens in our current homochirality, if built from molecules that have the exact opposite chirality, would essentially function the same, but be incompatible metabolically with our current
Yeah,
known
yeah,
forms of life.
yeah. This is all speculation. The speculation is that life began to kind of start to get formed when RNA was formed, because RNA is catalytic, meaning that RNA can catalyze some of the reactions necessary for the formation of life. And as it turned out, L RNA is catalytic, dRNA is not. So I think that was the first kind of shift towards the L enantiomer as opposed to D enantiomer.
So when you say the it was the D form of RNA was not catalytic, you meant it was not catalytic given the available substrates that it was being working on? Because
Yeah, yeah, yeah.
should it be, yeah, so there was almost maybe a slight prevalence of the L opposed to the D at the time that the first RNAs self-synthesize? Is that essentially what you're saying?
Well, you don't have to have a slight preference for
A
one
frequency,
to the other
yeah.
because if you start with same amounts of L or D, because L is also autocatalytic, right? Then L keeps making more of itself, D isn't.
Right, right.
So pretty soon L takes over.
Right. It's almost a sort of a biological analog of the whole question of the prevalence of matter as opposed to antimatter
Yeah,
from
well,
starting conditions. It seems isomorphic analytically.
yeah, it is all accidental, right? That that it all ended up being L. So, but it's also a little bit, we're kind of cheating a little bit too, because we're basically saying, what's the definition of L? L is an enantiomer that interacts biologically. So why do we have L? Because they interact biologically, right? It it kind of is a circular argument to some extent.
Sure.
That is, if you're really trying to talk about this chemically, you wouldn't use L or D, you would use R or S. Some L's are R, some L's are S. And so, in that sense, you uh, you over all of biology, you don't have uniformity that they're all R or they're all S. What uniformity you have is that they all the ones that interact with biological matter are called L.
So setting aside the terminological discordance between biological and chemical definition of chirality, um, there is no a priori reason that if one were to take all of the biologics that are in their current form and they were flipped to their opposite forms, there's no a priori reason that mirror image version of life should not work, is it?
Uh, I don't know for a fact. I don't know if Wissam knows and he can comment here or
I
not.
can tell you this: we've only found L chirality. We've
No, no, no.
also only occurring in nature. We've never found any biologic systems that are dechiral.
No,
Yes.
that's not the question though.
No,
The
no, I
question
understand. Here's
is
here's
in
what I was going
a,
to say. Yeah, here's what I was going to say. We also have only found it on space, asteroids, etc. They've all been L chiral.
yeah,
I just recently was reading about this. So there's a
but that's not
theory,
the question.
right? The question is, could you mirror them and would they work? And
So
so,
can you envision a universe in which all biological matter
right,
was
was
D
dechiral.
and not
And
L?
and I think the problem is we haven't done too much. I know some things have been synthesized in their dechirality. And like you said, there's a big debate about banning it because we don't know, like if you were to make some sort of mirrored life, our immune systems literally can't interact with them in theory. So it would go completely undetected, and we don't know what would happen if you introduce dechiral life in an L
But
chiral
that
world.
no, no, that's not the question.
No, no, I understand. Here's what I was gonna say. We don't know if it's possible because we've never had it. So
We
we don't
can,
know if there could be.
we can, right? We can create. D environments. And what happens is that it is going to be different, because if you change both of the conformations, it it doesn't necessarily lead to them being positioned in the same way as they were under L. So just because you reversed all of them, it doesn't mean that you're going to get the same interactions, because some of those inversions may cause them to move away from each other as opposed
But
to being next to each other.
Sam, we've never. The reason why I brought this up, we've never created an environment that's even meaningfully complex biologically of
Sure, we have.
Have we?
Yeah.
What's an example? I'm learning about this
No, I
for the
mean
first
we
time.
we we make D proteins all the time. We make left-handed DNA all the time, B DNA. So,
Oh, I
or
was
the
actually
Z
unaware
DNA.
of this.
So,
This is new for me. So
yeah,
I'm gonna go do some more reading.
yeah, you should. It's no, we've done quite a bit of research on D stuff. So,
Have we been able to make actual something that we quantify as living in dechirality?
well, we can't really make anything living in L either.
Fair, fair.
So,
Well, yeah, fair. And you couldn't even engineer something from L to D. It would be too complex.
yeah.
Yeah.
But the hypothetical D mirror image lives, um, as far as the concern about their interaction with L image, um, the molecularly, presumably, they wouldn't really be able to interact meaningfully, except maybe chaotically, right? Like in terms of being infected by a mirror image life form, it wouldn't. Its molecules would not interact with L molecules meaningfully, or if insofar as they interacted, it would be totally stochastic and indeterminate, and not be like an analog of an L and an L interacting.
Uh, I uh I wouldn't go. First of all, I don't really know the answer to the question, but I wouldn't go so far as there's no interactions, because we don't know, right? There may be some parts, there may be that the interaction between the molecules uh, is is not affected by the chirality of the molecule. The active site is such that either conformation can fit in there. So so because of that, I don't want to say all or every. What I want to say that it would be expected that a D conformation doesn't interact, but it's not a rule.
The example of, was it Olestra was the G-chirality
Yes,
oil?
yes,
Okay,
yes.
so there's an example of where there is an interaction between the D chirality molecule and an L chirality organism, but it's not a biological. Well, it's not a molecularly functional interaction of the molecule, the D molecule, with the L molecule. Um, it's a physical reaction as opposed to a chemical reaction by the organism, a physical reaction by the organism, rejecting it because of
Uh,
indigestibility.
no, that's chemical,
Okay,
because
physical chemical. I meant, yeah. Okay,
yeah, because a rejection has to deal with chemical electrostatic stuff, and It's non-compatibility in terms of you can't form hydrogen bonds or van der Waals interactions or anything. So,
Sure, sure.
right, yeah.
Yeah, I apologize for my slight scatteredness. I just woke up and I'm
No,
not quite wired
that's
yet.
fine. No, these are fine. Hi, Christina. Welcome to the stage. Oh, why did you go down? Come back up. Okay.
Yeah, I think there's times when we've used chirality to have things not interact or degrade in biological systems now that I think about it. So I'm not sure if there's been any things like when they use lipid shells, to bypass cell walls. I think they tend to use L, but I know there's a few applications. I'm going deep in my mind to when I was first learning about this, when we were looking at things like biomolecular engineering and using inorganic and organic compounds. We would use chiral organic compounds to sneak in inorganic compounds, like for example through a cell membrane. Um, and there are certainly effects, like when you look at cholesterol or something, like there are effects of them. It's just they're very different, right? Like even if it's insulating or things like that, and not being as reactive.
Yeah, Briar, thanks. That's what I was trying to get at there, that it's a different kind of interaction between a D and an L and a D and a D, let's say, or an L and an L.
I think Sam, isn't it Pepto Bismol that the chiral isomer is like exceedingly toxic, like insanely toxic? It's just that I forget if it's D or L that we get for Pepto Bismol. It's just that the chiral isomer, because of the energy levels in the reaction, I think it appears in like infinitesimally small numbers. I just remember when I was first learning. This is going back like 30 years ago to chemistry class, but that's the one I remember.
Yeah, I don't know about Pepto-Bismol, uh, but that first of all, I don't know what the active compound in Pepto-Bismol is, except that I know it has bismuth in it. But what the pepto part of it is, I don't know. So I can't really answer that. But uh, uh, that there are many drugs that are like that. That the other isomer is either inert or is toxic. In software organic lab, one of the labs that you do is synthesize peppermint oil, because the other enantiomer of it smells like rancid uh, nuts. It really smells bad. And so you are randomly assigned, the starting material. And so you make it. And so half the class is smelling great, like peppermint, and the other half, they're like, smells like shit. That really drives home the the chirality of the body, uh, and how chiral molecules interact differently. All right, I've been going uh uh for oh, Christina.
Sorry, I changed my mind. I this is somewhat back to what Otter and Briar were pointing to. Like, uh, if I because I don't know what I'm talking about. Okay, um, if I'm thinking about L chirality, and if I even think like I have a handle on this, um, then any living thing that has this, which we think all life on this planet does, right? Then that means even the oxygen and carbon dioxide process that happens and how oxygen happens with hydrogen to make water, right? So that anything you create with an R chirality, even if you could, how could it continue to exist if everything in this particular, let's say, planet is L chirality formed, whether it's a living being or processed through L chirality, that we would think that if we created R chirality, it could exist here, right? Because presumably the oxygen and the carbon dioxide and water and so on is not living in a vacuum either because it's being processed through these living beings. So yeah, I don't know. Does it, like, I feel like the idea of us being able to somehow create it, even if we could do L chirality, like that aside, even if we could do R chirality, like this feels like it wouldn't survive. What could it survive on? Or am I missing something?
Some molecules can't be chiral. If you like look at your hands, like your left and right hand, they're not exactly, but they're roughly mirror images. But there's no way, this is an easy way to kind of understand it, right? If you try and actually overlap your hands and have them in the same orientation, your left and right hand can't do that because they're mirror images. So there's no way of physically manipulating them. You'd have to completely deconstruct and rebuild. But a lot of um, I don't know if I'd say a lot, but at least a significant number of molecules out there aren't chiral because you have to have like some type of asymmetry. So although there is asymmetry in, like, for example, H2O, it can't be chiral because H2O, it has this thing with the hydrogen bond between the two hydrogens and the oxygen. So
No,
it
no,
has
no,
like,
no, 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 no.
well,
You went too
could
far.
too far. I mean, you can't have chiral water though, right?
That's
Because it
correct.
does. Yeah.
But
So
that doesn't have to do with
Right, but
hydrogen bonding.
this isn't
So
what I was
let's
saying. This wasn't
let's
what I was saying though. This
okay,
isn't
yeah,
this.
yeah. Let me take a stab at it. So first of all, as Breyer was saying, to be chiral, to be potential, to yeah, to be chiral, you need to have a central atom that's connected to four different things, or at least four different things. Why? Because for an object to be superimposable on its mirror image has to have an internal mirror plane. If you have four or more different groups attached to one atom, you can't have an internal mirror. In water, you can have an internal mirror that includes the plane of hydrogen, oxygen, hydrogen. And with the lone electron pairs on the oxygen are on different sides of the mirror, but are projected back onto each other. So that's why water is a symmetrical molecule. But if you look at the slides that we have, the amino acid, the central carbon has a hydrogen, has a side chain, has an amino group, has an acid group. Those are four different things. There's no way you can draw an internal mirror through this. So the the interactions that happen, right? If you are a chiral molecule, you you interact with other chiral molecules differently. That is, if both of you are left-handed, or one is left-handed, one is right-handed, those interactions are different. They lead to different outcomes. And so, am I on the right track in answering your question, Christine?
No, but
Okay.
I'm
So
just
why
gonna
don't
let it
you?
lie.
No. Go ahead and uh, expound on what you were saying, because I want your question to be answered.
I think that what I'm trying to get at is something we can't even point to exactly, because if most of our knowledge is from this particular planet and chirality, and specifically I'm thinking about like how DNA rotates, and that not rotates, but is like exists in that
Helical.
way.
Yeah.
Well, yeah, but to the left. Um, the point is that we don't know what ecosystems look like and how they produce what they produce with our chirality living beings, right? So we don't know what the chemical processes are going to look like. We don't know whether they're going to produce how we understand water, how we understand oxygen, right? So those are the chirality types of ideas that we don't even have an ability to study yet or understand yet. We don't know what they would produce. And so, like, we can talk about how we understand the chemicals and the chemistry of and the molecular structures of things on this planet, but that is within the context of chirality in beings, right? Plants, animals, and
Well,
so on. And so, like,
but we can create microenvironments in the lab. We can
but
we
hold on a second.
can
You said before we could not create our chirality because we couldn't even create chirality,
no
right?
that no the question was a life form
Right,
that's made out of D stuff and a life form. And I said, we haven't been
right,
able to make a living cell regardless of
right.
its chirality. So,
I
but
think I'm gonna let it
but
go.
that's
Thank you.
that's why I said we can make microenvironments, right? A single protein interacting with a single molecule, and we can see how those behave. We know we do have two other forms of DNA that have different helical structures, bDNA and zDNA. And so we can synthesize those in the lab and see how they interact with natural proteins and synthetic D proteins. And so we do create these microenvironments where we can study different reactions and then draw conclusions collectively from all of those. Though I agree with you that we really at the end of the day don't know, but I would disagree that we're as blind as you think we are.
I think, Christine, if I can venture to rearticulate your question, I think you're saying basically that um, that in order for our chirality to work, the inorganic molecules that that these molecules would have to interact with don't exist here on Earth. But the chirality of biologics, of organic chemistry, is different than the conformations of, say, oxygen, oxygen molecules or methane molecules. It's not that there's an absence of those inorganic substrates. It's that chirality doesn't matter in the same way for inorganic molecules as organic. Is that, Sam? Does that make sense? Do you see where I'm barking up?
I think I think my problem is that I can't follow the usage of some of the terms, meaning that when you use the term chirality,
Sure.
I'm not sure if you and I are using that term in the same context. And because are you by chirality, are you meaning like a certain enantiomer? Like we have the R enantiomer or the S enantiomer. Like
Yes.
so let's say we have one of the enantiomers in, let's say, our environment, and normally on Earth, this interacts with another R enantiomer. Now we're in a situation where the second compound is the S form, not the R form. So you're saying that are they going to interact? And the answer is most probably yes, they are. But it may or may not be the same interaction that the R&R had, right?
Right.
It could
Yeah,
be
that's the
a
point I was
different one.
making. I was trying to re-articulate Christina's point for her, um, which was the conformation of inorganic molecules like oxygen. She was saying if we did create an R version of these molecules, and we have in the lab, I think, Christina, and correct me if I'm wrong, I think you were saying that it wouldn't be able to interact with the inorganic molecules that would be required. Um,
No, that's not correct,
right, and
because
I think that's what Christine was
if
getting at. I think that's what Christine was getting at, is what I'm
yeah,
saying.
if a molecule is asymmetrical, then its interaction with ROS is going to be the same.
Right. So, Christina, does that answer your question now?
No, thank you.
I
We're
Hey,
tried.
doing all this. Never mind. Sorry.
so on the something Breyer said a while ago has kept me up at night. If well, not really. This has been daytime the whole time, but it's been this idea that so on Earth, all the amino acids and protein, all the stuff is left-handed, right? Um, and Breyer said a thing that even in asteroids, the amino acids we find are left-handed, and that was bothering me because I'm like, that would be astonishing if that were true. That would be like, okay, it's not just like a life thing on Earth; it's somehow the universe has decided that things will be left-handed. So that would be like I would hear all the intelligent design people talking about this. This would be like a marker of some grand design. So I looked it up, and it seems from my quick searching, it seems that's not true. That there is typically a 50-50 mixture of L and D amino acids when they find amino acids on extraterrestrial sources. So
That was my impression also, but again, because I didn't have the facts uh, at hand, I didn't say anything.
Ooh, I'm
But
gonna have to I'm gonna have to double check that, because if that's true,
yeah,
I was reading about this a while ago, and it was a theory about seeding of chiral elements, so could have been not true. I'm gonna double check
well, I mean,
myself.
we agree that it would be astonishing, right?
Oh yeah, yeah, yeah. It would indicate that there's something that actually favors like alchirality in the universe. That would be my interpretation. Or, all these theories about not um like you mentioned, like some sort of intelligent design, but just you've heard the bioseeding theories, right? That whatever is seeding a lot of the universe happens to be
Oh
L-chiral.
yeah, the life
okay.
started way
So
earlier
hold
than
on a second.
Earth. Yeah.
Instead of saying L-chirality, say the L-isomer or L-enantiomer. L-chirality doesn't mean anything. So chirality leads to L-isomer and D-isomer. Chirality is a concept. It's not a thing.
Thanks. It's
So
been
yeah.
literally 30 years since I studied this,
No,
so I
I know.
appreciate it.
I'm just trying to kind of if we're using the terms for everybody, so we're using the terms correctly.
The exception I found that there was some like ancient meteorites that were like dug up, and that there was a bit of a bias, but they still shared left. I'm just gonna say left. Well, L and D, left and right. Um, there was a bias. It was further away from 50-50 than one would have thought, but it wasn't that far away.
Yeah,
And yeah,
it looks like
yeah.
the Hayabusa, when they went to that Hayabusa mission, went to Ryukyu and took the samples. What I'm reading is those were 50-50. I'm kind of seeing the same thing as you, that there were. So now what scientists are saying is it might have been contamination on some of those early samples, why they showed up as disproportionately out, which would make sense.
And also, there may have
So
been biases. There may have been biases in the sampling protocols too. But the contamination is very likely. Yeah.
With that
So,
Japanese Hayabusa mission, it says it was almost exactly 50-50. So it does look like yeah, that it that bias is strictly towards Earth.
so you can get situations in chemistry where you react to achiral molecules that are not chiral with each other, and the product is chiral, uh, and it's not 50-50. So in these reactions, so on the left hand side, so let's say the new compound can either come from the right side or from the left side. If it comes from the right side, it makes the R enantiomer. If it comes from the left side, it makes the S enantiomer. Somehow, the arrangement of molecules in the rest of the compound is such that the approach to one of these sides is more strained, more conformationally crowded than on the other side. So most of them, most of the reaction takes place on one side rather than on the other. And so in these types of reactions, we calculate the EE, which is the enantiomeric excess of to figure out which enantiomer is favored more than the other. And so some of the theories of how life was formed kind of revolve around that one of these reactions had enantiomeric excess on one side. And so then that kind of perpetuated the whole thing so that the other ones didn't get involved.
Sam, I just found out something that might change all of us, and we might all have to admit we're wrong here. It turns out that there are some bacterial species that do have a substantial amount of their cell wall as being D isomers of certain amino acids. And the theory behind it is to avoid detection and natural, it's evolved this bias. So I was always taught on, just like we've all been saying, but it does look like there are bacteria that do have D isomers of certain amino acids, which is shocking to me. I did not think that was true, but I checked now 3 or 4 different sources and it appears
Oh,
to be true.
great. Yeah, I didn't know that.
Right? That totally throws me for a loop because I know the whole discussion that I think it was Otter brought up about, like, I a while ago watched a series of lectures on this, on how they're talking about proposing bans on lab development of D isomers because if they go out into nature, um, they bypass so much immune and other conditions that they might be able to just outcompete, because it's like throwing them out into nature with, the analogy for people who aren't familiar with this, would be like throwing out a animal that has no predator but also can't die from bacteria or viruses. So, but it looks like some viruses, of course, the theory would be evolutionarily have evolved such that their cell walls. Yeah, I didn't know this. This is really interesting. I'm going to be spending some time reading about this now.
And that ties into my earlier hypothesis that if there were, in that case, it was if there were, but now that we know that there are D isomers of these particular molecules that have analogs that have L isomer versions that are biologically active, that the D isomers would not be able to interact in the same way with the L isomers. So that's the competitive advantage of having a surface protein that's in the D form as opposed to the L, because the immune responses or receptor the detection, the molecule is essentially invisible to the normal L isomers that would normally detect a surface protein. It doesn't really trigger anything biologically or chemically. Well, biologically.
So Christina, as far as I know, solar radiations don't get involved in chirality. The prevailing theory for the handedness of DNA is what I said earlier, which is that RNA tends to be catalytic, well, only one isomer of it, and it is thought that it is both autocatalytic, that is, RNA makes other RNA, and also catalytic with respect to DNA. And it's because one enantiomer is, or one isomer is catalytic and the other one is not, you end up with the handedness of the DNA that we see today. Uh, Again, this is a theory, but it's a theory that led to a Nobel Prize. So I don't know. It's at least has some legs.
Tom, that example, I know you said you have to go. Do you have a moment, Tom, to explain that? And I'm sorry, Christine, I think I started at the same time as you.
I'm a bit skeptical of the claim when someone says they have to go and then they stick around for another 5 or 10 minutes. Um, I'm
Hi.
just saying people use that tool a lot to make space for themselves and then they stick around for hours. So, like,
Oh yeah, yeah. No, I was
but go
on
ahead.
the
I'll
way
give you the
to
benefit of the doubt.
bring my mom, pick my mom up. That's why. But I would, I'm talking on the go, and I'll be really quick. So there are two things that I heard. So D amino acids used for cell walls and other D amino acids preferentially used is a very interesting story in microbiology. We don't really know what's going on yet. It's all guesses and speculations. But there are enzymes out there that convert D to L or vice versa, and based on the preferences. So you could say that, well, why do they have these? So that's one package that I was going to say. I think is something for us to think about. The other one is I looked up Sam, and Sam is actually in intellectual property as well as a scientist. So the question that I would like to bring up, which I don't know the answer to, is very quick, is um, omeprazole is a drug that has R and S enantiomers, and its patent was running out. So the company, very quote unquote sneakily, um, isolated the S enantiomer and says this is better and got a new patent out of it. But chemically and also biologically and pharmacologically, I don't think there's any evidence to say the R and the S is any better than the other. But maybe in patent law they say, well, it is different, quote unquote, so you deserve a new patent. So that's, I think, is a rather fascinating use, convenient maybe, use of the patent law. And it will be interesting to hear what you have to say. So I will be listening while I'm on the go, but thank you very much.
So I don't remember the omeprazole case uh, very well. There was litigation over this, and I don't remember what the outcome was. It was a few years ago. But there's another compound, uh, Lexapro, and Lexapro and Celexa. Celexa is citalopram, and citalopram is a mixture of RNS uh, enantiomers, 50-50. And after Celexa was on the market, the pharma company figured out that it's actually the S isomer that's biologically active, not the R isomer. And so they went through a lot of trouble separating out the isomers, and they started marketing S-citalopram, which is Lexapro. And so the good thing about Lexapro was that you took half the dose, because what happened was that the R1 wasn't like completely biologically inert. It also exerted some biological activity, but those activities counted as the adverse side effects of Celexa. So when they switched to Lexapro, the dose went down by half, and the all of R was completely eliminated. So you basically started you the adverse event profile went down significantly, and so that's why they could get a patent on the S uh, isomer, because it really did have a different biological function than the racemic one, the one that had the mixture of both R and S. But omeprazole, I don't remember. Oh, Tom isn't even here. Okay, it's great. Ask a question and then leave. Right. Anybody else? I'm kind of getting tired. I've been at this for 2.5 hours. So I'm gonna stop this now.